In this lesson, I want to discuss the process known as excitation contraction coupling. Now, just to give you guys a little bit of an orientation, what I actually have in my little simple diagram here is a terminal cisternae. Remember, that is part of the sarcoplasmic reticulum. And here we have G-actin. So G-actin. So I'm going to erase that. And one thing I want to draw on G-actin is the active site of G-actin here. I want to color this in so that we can see the active site clearly, because this active site binds to myosin heads. And so that's one thing that's missing in this little simple diagram for a teaching lesson, to help you guys understand the process of excitation contraction coupling in muscle tissue. So our G-actin has our active site here. I'm going to erase my terminal cisternae, but I want to make a point that Ca2 plus is stored inside here. Now remember, with a muscle cell, when we have calciums in a high concentration in the terminal cisternae, a muscle's relaxed. So this is a resting muscle. So there's a few other things I need to do here. Number one, I'm going to draw a myosin filament here. Then I'm going to draw a myosin head. Now, myosin heads want to bind to G-actin. Now, I'd do this a little neater in a classroom, but the little myosin head wants to bind to G-actin. Here I gave you guys a little thing as if this were like a date, and myosin wants to hold G-actin's hand. But preventing this is a two-part major protein complex. I'm going to use red to draw one called tropomyosin. So tropomyosin is here. The little red dot's tropomyosin. And then I'm going to use blue to draw the three parts of the protein known as troponin. So troponin, tropomyosin complexes, they function to prevent myosin head binding to the active site of G-actin. So one of those things we knew that happened if I had the T-tubules sitting here, my T-tubules, and the action potential arrives into my T-tubules. When that happens, calcium, Ca2+, is released. When calcium is released, it will go towards our nice little troponin tropomyosin complex, and it will rotate it out of the way, and we'll move him over here. So now that he is out of the way, we now, and then you remove him so you can kind of see this, we now have him out of the way, and then we can form the cross bridge between myosin heads and G-actin, and this results in the coupling between these two enzymes. These essentially put myosin head functions like an enzyme. It is an ATPase. So ATP actually will have to leave the charged myosin heads, and ATP leaves, contraction occurs, the muscle then begins to contract. Now, when calcium gets pumped back in, and the calcium pumps engage, and calcium comes back into the cell, troponin tropomyosin complexes will go back the other way. And this will result in these guys having to let go. So ATP will go back in, become ADP, and inorganic phosphate. So I actually have to uncouple this. Draw my myosin head back in here. Let me remove the ATP, and it's back inside the highly charged myosin head. And then we draw back troponin. Here's tropomyosin in red, the three parts of troponin. And this, as it's back in its conformation, this is now relaxed. Calcium is back in a high concentration in the terminal cisternae. It's relaxed. If it's in a low concentration, it's contracted. And this is essentially how we work these sarcomeres, where we have our thick filaments here and our thin filament there. And I know that I made these a little bigger, but just for safety's sake, this is a little easier to see. But I hope my little quick teaching lesson on excitation, contraction, coupling mechanism was useful in you understanding muscle cell physiology, muscle tissue physiology. And this concludes my little video on excitation, contraction, coupling. Thank you.